Assalamualaikum warahmatullahi wabarakatuh. Today we're still in chapter 3, photosynthesis. So for this part, we will learn about light-independent reaction. As our previous video, we have been learned about the light-dependent reaction. Okay, because for the process of photosynthesis, they have two major reactions, which is first is regarding to light-dependent reaction. Okay, under light-dependent reaction, okay, we learn that they have two different process, which is we call as cyclic or non-cyclic reaction. Then we have light independent reaction. So this part we will learn today. So this light independent reaction we can call also as a Kelvin reaction. Alright, so in this slide you can see that what happened in the process of photosynthesis, and you know that this process occurs in the chloroplast. So as you can see there, for the light reaction and for the Kelvin cycle, they are illustrated, okay, in the chloroplast. However, for these two different processes, they happen in different areas. Okay, which is for the light reaction, you can see they occur in the green color part, which is this green color is the thylakoid membrane. Alright, so for the light reaction, you can see that, okay, in our slide also we have compared between light reaction and Kelvin reaction. Alright, so we go first for the light one. So in the light reaction, we know that, okay, they use the water, need a light because they are dependent reaction. The process of light reaction, they require the photo system, okay, which is PS1 and PS2. So you need to recall back what is non-cyclic light, what is the cyclic pathway. And the process, they will be produce ATP and NADPH. As their byproduct, they will produce the oxygen. Right? Now we go to the Kelvin cycle. Okay, for the Kelvin cycle, they do not occur in the thylakoid membrane. However, they occur in the stroma. Okay, as I mentioned to you in your previous slide, okay, this stroma, if you can't remember what actually the stroma is, so they actually is like a cytoplasm in the cell. But here we talk about the organelle that you call as a chloroplast. So their cytoplasm, we call the name as the stroma. Alright, so the stroma, where the process of the Kelvin cycle takes place. And you can see that they require the carbon dioxide, CO2, okay, and they produce G3P. What they use? They use the ATP and NADPH that produce from the light reaction. Means this light reaction, they provide the ATP and also an ADPH, okay, to use by Kelvin cycle. Alright, and this Kelvin cycle will be return the NADP plus and NADP and PI to the light reaction. So I hope all of you can see these differences, okay, between these two reactions, okay, light reaction and Kelvin cycle. Okay, now we will talk about the Kelvin cycle. Okay, for the Kelvin cycle, they have the processes. Okay, so you need to remember they have four processes, or we call as four phases. Okay, so now we look on the phase 1. Okay, so for the phase 1, we call as carbon fixation. After I explain to you regarding to phase 1 until phase 4, so you may look on the illustration regarding to the cycle. Okay, But now we go one by one explanation, okay, regarding to the phase. Alright, so in the first step of phase 1, okay, in carbon fixation, where in this process, carbon dioxide, because you can see in the previous slide, they require the CO2. So because of that, in the first process, that CO2 will be attached to the 5-carbon sugar. Okay, we call as ribulose biphosphate. Or their famous name or short name, we call only as the RUBP. Okay, so when the CO2 attached to the RUBP, okay, it will be immediately produce 6 carbon compound. So this process have been catalyzed by the Rubisco, or we call as RUBP carboxylase. Means the CO2 attached to the ribulose biphosphate by who or do by what kinds of the enzyme? So that enzyme is the RUBP carboxylase, or we call also as their name Rubisco. So this attachment between CO2 to the RUBP, so they produce the product, which is that product is 6 carbon compound, but they actually have unstable form. So because of that, the 6 carbon compound will be immediately split in half. So they produce 2 molecules of 3-phosphoglycerate. Okay, now we are in phase 2. So phase 2 we call as reduction. So remember, in our last or in our first process, which is carbon fixation, we get the 3-phosphoglycerate. So because of that, in reduction, each molecule of 3-phosphoglycerate will be receive one phosphate group from the ATP. Means in this process reduction, they require the ATP from the light reaction. So when these 3-phosphoglycerate receive one phosphate group, so they will become 1,3-bisphosphoglycerate. Okay. In this process also, they use NADPH from the light reaction. So this NADPH will be donate their electron and reduce 1,3-bisphosphoglycerate to a new simple sugar that we call as G3P. Remember, for G3P, you have been found it in the glycolysis, which is their full name is the glyceraldehyde 3-phosphate. And remember, in the process of a Kelvin cycle, you can see that for every 3 molecules of CO2, so there are 6 molecules of G3P. Okay, this means in every cycle, in single cycle of Kelvin cycle, they require they require the entering 3 molecules of CO2. So because of that, at last, they will produce 6 molecules of G3P. So from this 6 molecule of G3P, so one of the 6 molecule of G3P will be used in order to produce the sugar, which is the glucose. And another 5 molecule of G3P will be needed to regenerate the RUBP. Okay, now in phase 3, So in phase 3, we call as regeneration of the CO2 acceptor. Who is the CO2 acceptor? Or who is the compound or sugar that accept the CO2? Of course, is the RUBP. Okay, so means here the process of phase 3, we want to regenerate back the RUBP. So in this process, okay, the carbon skeleton of 5 molecules of G3P will be rearranged in order to produce 3 molecules of RUBP. Remember, in the phase 2, they produce 6 G3P. One will be used to synthesize the sugar. And we have a balanced 6 molecules of G3P. So this balanced 5 molecules of G3P will be enter the phase 3 for regenerate the RUBP. So how many molecule of RUBP they able to produce? From 5 molecule of G3P, which is they able to produce 3 molecule of RUBP. So in this process, they also require the ATP. So when the RUBP is regenerated, so now RUBP is ready to receive another CO2 and makes the Calvin cycle continues. Okay, now after that, in the final phase, we call as the product synthesis phase. So the product synthesis phase is regarding to that one G3P, okay, from 6 molecule of G3P will be used to synthesize the glucose and other carbohydrate. Means they also can produce not only the glucose but another sugar. Okay, so that's all for the process of phases in the Calvin cycle. So you need to remember they have 4 phases and you're able to explain. Okay, now if you can't imagine, okay, what is the carbon fixation, reduction, and then regeneration of the RUBP, then synthesis of product, so you can see here. So in this slide, they explore to you, they extract to you regarding to the Calvin cycle illustration, where you can see for the phase 1, as I told you, the name is the carbon fixation, means fix the carbon to the simple sugar. What that simple sugar? That is the RUBP. So in this process, you can see 3 CO2. Okay, 3 CO2 need to begin the Calvin cycle or to start the Calvin cycle. So that CO2 bind to the RUBP, okay, by the Rubisco and form the intermediate 6 carbon compound. So if you can see that, okay, from 3 ribulose biphosphate, RUBP have 5 carbon compound. Okay, 5 carbon. So now when the CO2 bind, so it will produce 6 carbon compound. Okay, the 6 carbon compound is unstable. So because of that, it will be split in half. So from 3 6 carbon compound, when they are split into, so they will produce 6 3-phosphoglycerate. Why 6-phosphoglycerate? Okay, commonly for the student, they always confuse, okay, for this type. Okay, how actually from the short intermediate product, they able to produce from 3, they able to produce 6. Okay, 3-phosphoglycerate. So if you can see there, okay, for the 3 short intermediate product, 6 carbon compound, they have how many total for their carbon? They have carbon, they have 6. Okay, so 6 times with 3, total carbon we got 18. Okay, so in order from the 18 carbon compound, they produce 3 phosphoglycerate. So how many 3 phosphoglycerate they able to produce from 18 carbon compound? Okay, that origin from the 6 carbon compound intermediate product from the RUBP bind with the CO2. Of course, is 6. So they produce 6 3-phosphoglycerate. So if you still blur or not understand, so you can count. For example, here 3-phosphoglycerate, they have actually 3 carbon compound. So this 3 carbon compound you times with 6 molecule of 3-phosphoglycerate. So how many you get? Carbon you may get in total also is 18. So that's correct. So from the short intermediate product 6 carbon compound, okay, so they able to split into half. So they able to produce 6 3-phosphoglycerate. Okay, then we enter for the phase 2, which is in the phase 2. Okay, we need to use the product from the light reaction, which is in the reduction we use ADP, we use the NADPH. So how this process occur? This 3-phosphoglycerate receive the receive the phosphate from the ADP and become 3-phosphoglycerate. So this 1,3-bisphosphoglycerate then reduced by the NADPH. So become the 6-glyceraldehyde 3-phosphate. Okay, so this 6 glyceride 3-phosphate G3P. Okay, then one molecule, only one, remember, only one molecule of G3P will be used to produce the sugar. And another balance 5 molecules of G3P will be enter the phase 3, which is their purpose to regenerate the RUBP. So that 5 molecules of G3P will be rearranged and become 3 RUBP. Okay, so now this 3 RUBP is ready to receive another CO2 to continue the Calvin cycle. And they enter the phase 4, which is product synthesis. Okay, so this is focused on that one molecule of G3P is or will be produced a glucose or another sugar. Okay, so that is all about the Calvin cycle. Okay, when you look on their all the simple sugar that involve and also their phases 1 until 4. Alright, so now is about the summary of process, okay, in the light dependent reaction, and also a summary of process in carbon fixation reaction. So if you look on that, they are focusing on what the material that they need and what the end product they have been produced. Okay, for example, for the light dependent reaction means the light reaction. So of course the energy they get from the sunlight. So remember, for the light reaction, they use the sunlight to split the water. Okay, so they able to get the oxygen and they manufacture the ADP and reduce NADP plus to NADPH because they produce ADP and NADPH. Okay, now under the light dependent reaction, they give a summary regarding to the photochemical reaction, okay, electron transport, and also the chemiosmosis. Okay, photochemical reaction actually is the process involve the photo system. Okay, so happen in the photosystem. So because of that, for the summary of the process, the chlorophyll is activated. Okay, because they involve the chlorophyll A and all the accessory pigment. Okay, they also involve the reaction center, give up photoexcited electron to the electron acceptor. This is regarding to when the electron, okay, of the chlorophyll A receive the energy, photon energy from the accessory pigment that they get from the light. So that electron will be excited to the primary electron acceptor. Okay, so what the needed material that they need? Light energy. Okay, for the photochemical reaction. What else they need? They need the pigment. So if you write about pigment, you put their answer is, for example, chlorophyll A and accessory pigment. They also, that answer also correct. Okay, so the end product from this is electron. Okay, now we look on electron transport. So in this light-dependent reaction, you remember in the process of non-cyclic and cyclic, they involve the electron transport. So when you want to summarize about the electron transport, they actually the process of you transport the electron, okay, along chain of the electron acceptor. Of course, when you are transport the electron, you require the electron acceptor, which is in the light reaction. What the types of the electron acceptor they use in the electron transport, which is PQ, plastoquinone, cytochrome complex, and PC, which is plastocyanine. Okay, and then they need to have the ferridoxine and also an ATP plus reductase. Alright, so all of these, okay, will be, for example, here electron reduce NADP plus. So means in this NADP plus will be received the hydrogen ion get from the splitting of the water that accumulate in the thylakoid space. What the needed material in the electron transport is the electron and ADP plus, the water, and also electron acceptor. So of course, end product from the electron transport, they get the ADP. If you can remember the reaction of light of the cyclic and of the non-cyclic, they produce also the NADPH. Then last in the light reaction, they have the chemiosmosis. So in the chemiosmosis, all about the hydrogen ion gradient and about the ADP synthase. Okay, so because of that, the needed material in the chemiosmosis is the proton gradient and an ADP plus with the PI, inorganic phosphate, because their end product is the ATP. Okay. Now we look on the light independent reaction, or we call as Kelvin cycle. So we do the summarize regarding to carbon fixation reaction. Okay, so why we focus on the carbon fixation reaction in the Kelvin cycle? Okay, because all the question, all the discussion regarding to the Kelvin cycle, regarding to the photorespiration, C4, C3, C5 plant, all about their carbon fixation. Okay, so in this carbon fixation, CO2 is used to make the carbohydrate. That is a very general summary. From the CO2, they produce the carbohydrate. So what the needed material for the carbon fixation is the first compound, which is RUBP, or their name is ribulose biphosphate. Okay, they require CO2, ATP, and NADPH. Okay, from the light reaction and all the enzymes that involved in the Kelvin cycle. Okay, what the end product that they produce? Okay, they produce a carbohydrate, which is the sugar, or in the general reaction of the photosynthesis, they produce the glucose. Okay, they also produce the ADP plus inorganic phosphate and also an ADP plus NADP plus, because they use the ATP and NADPH, so they will produce or regenerate back the ADP plus PI and also the NADP plus, and give these two back to the light dependent reaction. So that's all for this